Ladies and gentlemen, my name is Tom Sauer and I'm here to introduce our speaker today. Our speaker is Britt Moore. Britt is a PhD student here in the department. He got his bachelor's degree at Truman State in Kirksville, Missouri in 2007 and then came here to do a Master's of Science with Tom Casper in National Lab for the Ag and the Environment, formerly was known as the National Soil Test. Yes. Then he went and taught high school environmental science for a year in Florida, and apparently that was a interesting experience. And he decided to come back to Iowa and work for Extension. And his area of interest is cover crops. So his master's degree was in cover crops, and then his extension work is in cover crops, and now he's doing a PhD here in cover crop effects on soil properties. So the title of his presentation today is, Are Rye Cover Crops an Effective Tool to Improve Soil and Water Management in Iowa? Welcome, Britt.
Hello everyone, thank you all for coming. Today I would like to talk a little bit about some of my dissertation work. As Dr. Sauer mentioned, my dissertation work focuses on whether or not cover crops can be used to improve soil and water management in Iowa. So I'll be talking a little bit about some of the preliminary data I have, but it's important to stress that I don't have all of my data completed yet. So everything that you see here today, data-wise, will just be from 2015 and 2016. So just preliminary data. So first of all, I'd like to start by talking about why we care about soil and water management in the first place. Water management is a really hot topic lately, in part because of the increased focus on yields. So there's a lot of talk about increasing crop yield, and there's been a lot of focus on improved genetics and other things that can get us to higher crop yields. But what I'd like to preface before I start my talk is that without water, genetics can only go so far, right? So if we think about having increased yield, a bigger ear of corn, in order to have that larger ear, we need a sturdy stalk. We need leaves that are going to be transpiring water, so that yield increase is not going to happen unless we're mindful about how the crop is using water and where that water will be coming from. So I'd like to start by talking about soil as a natural resource. Soil erosion is a major problem in Iowa, as many of us know. Conservative estimates for soil erosion in Iowa are about 5.5 tons per acre per year. And I say conservative because these measurements are from the Iowa Daily Erosion Project and NRI data. They only measure sheet and rill erosion. They don't measure any gully or stream bank erosion. So 5.5 tons per acre per year loss is a very conservative figure. In actuality, we're probably losing a lot more. When we think about soil, we really should treat it as a non-renewable resource. At least it's non-renewable over a human lifespan. Estimates for this part of the world are about 400 years to form 1 inch of topsoil. Any of us that have spent any amount of time in Iowa, especially in the spring, when we have a big rainstorm and we have fields that are not covered, then how long does it take to wash away 1 inch of soil? It can happen in a matter of seconds, minutes, hours. So we really need to be mindful about protecting our soil resources because erosion is a major problem. So I really like this figure because it kind of drives the message home. For every pound of corn that is produced in Iowa, we are losing 1.2 pounds of soil to erosion due to our cropping management practices, right? So that's a huge, huge loss, and it's a rate of soil loss that will not be sustainable over the long term. So if we think about soil loss, since Iowa was settled by Europeans and since cultivation began, we can see that there's been a drastic reduction in soil organic carbon. So this data, it is unpublished data, but it came from Dr. Laird's lab, and they did a study where they looked at several sites around the state of Iowa, and they compared prairie sites, so sites that had never been cultivated, to sites that were on the other side of the fence row that had been cultivated. And what they found is that the average difference in soil organic carbon loss was 42%. So since Iowa was settled and since cultivation began, estimated that we have 42% loss of soil organic carbon. Again, these rates of carbon loss and these rates of soil loss are not sustainable over the long term. So we really need to be thinking about, are there effective management strategies that can help us mitigate some of this soil degradation? So I want to shift gears a little bit and talk about water. So we've already talked about soil loss and soil erosion and degradation being a major issue, but I want to talk a little bit about water briefly. So we all know, and we've known since grade school, that the earth is composed mostly of water, right? About 3/4 of the earth's surface is water. What's a little bit less realized by most people is that the amount of fresh water on the earth's surface is actually relatively small. So estimates are about 2.5% of all earth's water is fresh water. Of that fresh water, an even smaller amount is water that can be readily utilized. So if we consider the water that's not stored deep underground or the water that's not frozen, then we're left with a relatively small amount of water that's left over for human consumption and agricultural use. We can see here that soil is actually a very significant reservoir of fresh water. If you combined all of the water in the atmosphere, all of the water in Earth's biota, all of the water in the rivers, and all of the water in the wetlands, you still would not have as much water as is inhaled in soil. So soil is a major reservoir for fresh water. So shifting gears again to talk about agricultural land, this slide illustrates that there is an uneven distribution of agricultural land. But what's really interesting, I want you to keep this slide in mind, because going to the next slide, if we look at water distribution, so we've already talked about water as being a relatively scarce resource. But the problem is compounded because water is not evenly distributed. So we have the climate moisture index, which we can see in the map above, and that's a measure of the potential, the climatic potential for evapotranspiration to exceed precipitation. We can see that in many of the regions from the slide before, where the world's most important cropland is, and also in regions that have a very high population density, that the climate moisture index is actually very problematic. So we can see this band here. There's a lot of people living in that area, but water that comes from precipitation is unreliable. It's highly variable. We can also see from the map below that this coefficient of variation, which is an easy way to say that is just, it's a measure of how variable water is over a given year. we can see that that variability is also very high in areas that we rely on for cropland and areas that have a high population density. So the problem of water is compounded by relative scarcity, but also uneven distributions and high year-to-year variability. So why do we care about water? Well, going back to what I said at the opening of the talk, water is absolutely essential for yields, right? This is not new information. We've known this for a long time, but it's useful to keep in mind that for about every 10 bushel of grain yield increase, we need about 1 more inch of water. So again, if we want to move our agriculture towards high production, and people are talking about 300 bushel per acre corn, that's not going to happen unless we address the water issue. So, water is important, we know that, but we run into problems when we have too much water or too little water. The illustration here shows that if we start about 1930 up to current times, yield has gone up, which is what we expect. But if we look at years where the yield has been significantly lower than the average, almost all of those coincide with a flood or a drought event. So times when we had either too much water or too little water. It's made a major impact on crop grain yields. So, in short, soil and water resources are very important. This is important to keep in mind because the relative amount of the Earth's land that is arable or readily available for cropland is relatively scarce. And on that land, soil degradation is a significant issue. Right now, we're at a unique time in human history where we have a very large population that is still growing. We have a population, a global population, that's becoming a lot more affluent. So we have a population that has an increased need for food and an increased demand for higher quality products, such as meat. So what that is doing is in turn driving a larger demand on cropland, which is resulting in more soil degradation, more soil use, more water use, and more water demand. This is all happening while the stocks of these resources are going down. So we have increased demand and increased use while the stock is going down. It's going to create a major problem unless we can find strategies for sustainable intensification of agriculture. And what sustainable intensification is us finding a way to do more with our resources and to do more with agriculture while using less resources. So, the question at hand is that, are there agricultural management practices that can help us deal with soil and water issues simultaneously? So we've already talked about soil degradation being a problem, water scarcity is a problem. What, if any, practices can be used to deal with these problems simultaneously? To answer this question, I'd like to go back to the work that I did for my master's project with Tom Casper over at USDA Inlay. So starting in about 2009, we decided to look at whether or not rye cover crops are an effective way to improve soil quality. So we did our research at the Boyd Research Farm, which is located just in the next county over in Boone County, Iowa. And we had treatments of a rye cover crop following corn silage. we had it following corn grain, and we had it following soybeans, and various combinations of those different treatments. So we drilled the rye in the fall, and then the rye would be killed in the spring before the main crop was planted. So for those of us that may not be familiar with how a winter cover crop works, you typically plant the cover crop in the fall after the main crop has been harvested, and a crop like rye will overwinter. So the cold won't kill it. It'll resume growth in the spring. You'll kill it in the spring. For our treatments, we spray them with glyphosate. We kill them in the spring. You kill it typically about 2 to 3 weeks before you plant the main crop, so that there's not any competition with your cover crop and your main crop. So that's what we did for this study. We looked at these different treatments. This is a photo of us drilling in our cover crop in the fall. So you can see that this cover crop has been drilled after a corn silage harvest. And this is a photo of the cover crop residue in the spring after our main crop has emerged. This photo is important because we can see that there's quite a significant amount of cover crop residue left over from the cover crop growth. So all of the brown that you're seeing in the soybean interrows is cover crop residue. So what we found in that study is that when we averaged the treatments over our research fields and over both years, we found that our soil quality indicators that we measured, which was total organic carbon, particulate organic matter, and nitrogen potential mineralization, we found that all of these soil quality indicators were higher when a cover crop was present. So we had our soil organic matter, we had our particulate organic matter. Now, a lot of us are familiar with organic matter, but for those of us that may not be familiar with what particulate organic matter is a type of organic matter. It's technically defined as the organic matter between 53 microns and 200 microns mean diameter. It's not important that you remember that. What is important is that you know that POM is a variable pool of organic matter, and it's useful because it acts as an indicator of management-induced changes to organic carbon. So people like to use this POM measurement because it tells us something about how our land management is affecting our soil organic matter. We also looked at nitrogen mineralization potential, which is a useful indicator on how well the soil is able to provide soil organic nitrogen. So we looked at all these soil quality indicators, and we found that there was a significant effect when we had a rye cover crop following our main crop every year, and following years where we only planted a rye cover crop after our corn silage harvest. We also found that cover crop differences in the uppermost soil profile layer, so the 0 to 5 centimeter layer, was where we were seeing our most significant differences. For this study, we looked at 0 to 5 centimeters and 5 to 10 centimeters, but in the 5 to 10 centimeter range, we didn't find any significant differences. But for each soil quality indicator, we did find differences at the 0 to 5 centimeter range. So we found that soil organic carbon and particulate organic carbon were higher in plots with a rye cover crop than with a controlled treatment. We attributed this difference to the rye having more biomass in these treatments. So we had the rye cover crop following both crop treatments, but we also saw a significant difference when rye followed corn silage. For those of us that may not be familiar with how silage harvest works, you typically harvest for silage about 3 weeks before you would harvest crop grain yield. So we suspect that 3 extra weeks of rye cover crop growth in the fall gave that cover crop a lot more time to grow and to establish biomass. So crops in that treatment cover crops in those treatments had longer to grow in the in the fall, and they were able to put on more biomass. So we suspected that is why we saw higher soil organic carbon and particulate organic carbon in those plots that had the rye cover crop following each main crop and following a corn silage crop. We also found, as I mentioned a little bit earlier, that the effects of a rye cover crop were most pronounced in the uppermost soil profile layer. Again, we attributed those differences not only to the rye shoot growth and residue, which there was a lot of, but also to rye root growth. When you had the rye cover crop, there was a significant amount of root growth because you had the cover crop growing there compared to fields that did not have a cover crop. And also we found that there was no negative treatment effect on main crop yield, which is really important because one of the main criticisms that's been lobbed against cover crops is that people are reluctant to plant cover crops because they think that it may interfere with their main crop yields. Our study found that there was no significant difference one way or another. So even though soil quality was increased, we didn't find an increase in yields, but we also importantly did not find a decrease in our main crop yields. And this paper was published in the Soil Science Society of America Journal. If any of you are interested in the paper, I can send you a link if you come see me after the talk. So that was the initial study. Our study found that rye cover crops can increase soil quality, but other studies that have been done at Inlay, at ISU, and other places have also found that rye cover crops are an effective way to reduce soil erosion. They're an effective way to deal with nutrient leaching, and they fit in well with our existing corn and soybean rotations. But what about plant available water? That is the question that we really want to answer. As Dr. Sauer mentioned in his introduction earlier, after we completed the project I talked about before, I took some time away from academia and I took a job as a cover crops person for ISU Extension and Outreach. I would spend a lot of time traveling around the state of Iowa talking to farmers about cover crops. One of the things that I heard time and time again while traveling and talking to people about cover crops is, what about water? What is the cover crop going to do to my water? Is it going to be stealing water away from my main crops? Am I going to have a problem with water? It came up time and time again throughout my time in Extension. So it really got me thinking about, well, what is the cover crop going to do to water? So under the guidance of Tom Sauer and Rick Cruz, under their encouragement, I decided to start a PhD project trying to answer that very question. What is the rye cover crop doing with plant available water? So our research found that the rye cover crop is effective for enhancing soil organic carbon, but there's other research, notably research by Dr. Chanu and Dr. Hudson, that shows that soil organic carbon can significantly increase plant available water. So we decided to put those two projects together. We said that, okay, in our work here at Iowa, we found that soil organic carbon is increased. Other researchers are finding that soil organic carbon increases plant available water. What will happen if we look at rye cover crops as they affect plant available water? Because we know that we already have this increase in soil organic carbon. So we decided to take a closer look at that. And the hypothesis for the current project that we're working on is that rye cover crops will increase organic carbon, they'll improve soil structure, and this will lead to an improvement of plant available water. So we decided to look at the cover crop treatment effects on soil water retention. We decided to look at soil organic pools and see which of those will affect soil water retention. And we also decided to look at the relationship between soil structure, organic matter, and plant available water. So for our current project, it's at the same sites that we used for the previous study. The same treatments are in place. These treatments have been in place for a long time now. The Rye Cover Crop has been in place since 2001, and these fields have also been in no-till since 2001. So it's a long-term study, and for our current project, which began in 2015, we are looking at the Rye Cover Crops effects on soil physical properties and soil water retention. This time, however, we have made some changes. In addition to looking at different soil physical properties and hydraulic properties, we're only sampling in the top 5 centimeters. You'll remember from the study I talked about before that 0 to 5 was the only area of the soil profile where we found significant treatment differences. So this time around, we decided to focus on that uppermost soil profile layer. This time, we also limited our treatments to 2 treatments. We have a control treatment, which is no cover crop, and then we have a cover crop following corn silage and soybean. Remember earlier from the project before that we found that our soil organic carbon and particulate organic carbon differences were most pronounced in fields that had rye cover crops following corn silage, which we attributed to the rye having more time to establish and grow in the fall. So those are the treatments that we decided to use for the current study. So our 2015 and 2016 data has shown that total organic carbon is significantly higher in fields that have a rye cover crop treatment, which is very much in line with the results that we found in our previous study. We also found that particulate organic matter is significantly higher when a rye cover crop treatment is present. Again, this meshes well with the study that we did before, which showed that same pattern. This time, we also decided to look at macro aggregates, and we found that macro aggregates are higher in fields that have a rye cover crop treatment compared to fields that do not have a cover crop treatment. It's very important to keep in mind that there's only limited inference we can make from this data because it is only 2015 and 2016 data. We still do have more data to process, but the preliminary data so far looks promising. If we think about corn grain yields, so in the study that I talked about previously, the project that led up to the project that we're doing now, there were no significant crop yield differences. And so far, that trend is also holding true. There has been no negative treatment effect on main crop yield. If you look at 2015 corn grain yields, we can see here that there is a slight numeric difference, but there is not a statistically significant difference. Actually, just looking at the numbers, our cover crop treatments had a slightly higher corn grain yield than our non-cover crop treatments. And both of those treatments actually did better than the Boone County average for that year, which was only 192 bushels per acre. So that part is promising as well because, again, the results from our experiment can only go so far if we're showing a significant yield decline. But so far, that hasn't been a problem. So all of the properties that we have tested for so far have shown a positive rye cover crop treatment effect. The Rye Cover Crop has increased our soil organic matter and our particulate organic matter, and it has also increased our water stable soil macro aggregates compared to our control treatments. However, as I mentioned earlier, we still have a lot more that we need to do. So the next steps of this research will involve determination of our soil moisture release curves, and it will also involve fractionating the organic matter into different pools. So we know that the organic matter has increased, but what I'm really interested in finding out is whether there are different pools of organic matter that are more responsible than others for any potential increases or differences that we may see in soil water retention. So to wrap it all up, soil and water resources are in a state of peril, and our demand on those resources is increasing, and that's not going to change anytime soon. So we really need to be thinking about strategies for sustainable intensification. Sustainable intensification is not going to happen unless we come up with management practices that are able to deal with preservation of our soil and water resources. My research investigates whether or not rye cover crops are an effective way to move us towards that sustainable intensification. The preliminary data so far shows promise, but again, we have to take it with a grain of salt because there's still a lot more tests that needs to be run, and we only have 2 years of data so far. So with that, I would like to thank you all for listening, and I'd like to take any questions that you may have.
Britt, nice presentation. I noticed you saw the average yields were higher in those cover crop plots than the county average. Could you explain why you think that is when, you know, a lot of farmers that I hear from, they see a yield drag from cover crops?
The first thing that I thought about when I looked at this data was the fact that these plots have been in no-till since 2001. I think that can explain at least some of the difference. Sometimes oftentimes there is a yield drag when you transition into no-till, but these plots have been in no-till since 2001. So having that better soil structure, having that organic matter that has had time to develop, having increased biologic activity. There's actually a paper that just got published by Shipatello and Tom Casper that talks about earthworms. And there's plenty of earthworms out at these sites. So a lot of biological activity, no-till, I think that explains at least some of the differences that we're seeing.
So if a farmer asked you, you'd just tell them to hang on for 5 years?
That is what I would tell them, because no-till, I mean, it does have its problems. It's definitely a different management style and different skills involved in that. But when no-till systems are developed, and especially when those no-till systems are paired with a well-managed cover crop, then there is a potential to see significant soil quality improvements. Thank you for your question. Dave.
So I'm going to veer into shaky waters and not self-service. But this was kind of brought up last week when they talked about artifacts that have been design or even taking samples. So when you take a sample just down to Greenwich and you know you've got Ryman's there, how do you know what you're measuring is the type of carbon that you want it to be? That makes sense.
Yeah.
So I think Dr. Thompson brought this up last week. And is that okay? So if these things are experimental artifacts, but they show positive results, where does that take us?
Thank you for your question. I've tried as much as possible to be consistent. So when we're doing the carbon analysis, every effort is taken to remove obvious signs of root activity. Now, I'm sure that's not a perfect process, but when possible, there's attempts made to remove large pieces of organic matter. because those will have an impact on the final number.
So is there something to the different age of carbon in there, right? So again, is that a bad thing? There's this gradient, there's this temporal gradient of root age below
Mhm.
ground, right?
Right.
Where does that leave us? I know that's super broad. I'm just kind of curious more than anything else.
Yeah, so I'm not sure I know how to answer.
That's an open question to, I thought this was the audience that might answer that.
Yeah, Dave, I'm not sure how to answer your question. When we do take the undisturbed soil cores, we take them late May and early June. And the reason that we take them during that time frame is because it's a time of year where the previous cover crop roots have had some time to decompose, but it's also before the main crop has had time to send a lot of those roots into the interrow. So we are sampling at a time of year where there's not likely to be as much root growth in the interrow as it would be if we were to sample later in the summer or earlier in the spring when that cover crop is present.
Thank you.
Carolina.
Hi, great presentation. I have a question regarding why you hold on into 0 to 5 centimeters for your soil water measurements.
Mhm.
Like for soil carbon, there are some confounding reasons in the result that might be like by methodological, the way that you measure carbon, right?
Mhm.
But regarding the soil water, I'm wondering if you don't hold to 0 to 5 centimeters, there are other, like, and you look below that,
Mhm.
there might not be affecting like abiotic factors to the soil water content of your samples. So will you be able to go deeper or like look into different depths?
So for this study, we decided not to go deeper. And again, some of those reasons had to do with the fact that in the previous study, we did find that a lot of the soil quality differences were kind of restricted to that uppermost layer. And for my study, I do want to look at how those carbon differences relate to differences in plant available water. So I'm trying to keep it consistent by doing all of the measurements in the uppermost profile layer. But as you mentioned, there could be some value to looking deeper in the soil profile, but for this study, we're just restricting it to that uppermost layer. Dr. Laird.
Yeah, so I know your long-term goal here is to address plant water use efficiency. So would you tell us how you just will define plant water use efficiency and then how you intend to measure it?
Yeah, so with water use efficiency, as you can see, like with this presentation, I did not bring up water use efficiency because those waters can get muddy pretty quickly. So, I didn't talk about that. Long term, it will be difficult for us to measure water use efficiency. I think that's an important thing to know about, but our study does not look at evapotranspiration directly. So it would be hard to measure water use efficiency. And I guess to answer your previous question, I would define water use efficiency as biomass, the amount of plant biomass per unit of water. But without us measuring evapotranspiration directly, I can't say that our study is really going to look at water use efficiency. That's why I decided to go with the term water management, because we will be looking at plant available water, but we won't be looking at water use efficiency. And for us to look at the plant available water, then we just need to know the field capacity and the permanent wilting point, and then we can correlate that with the soil organic carbon measurements to see if there's any differences. But as far as water use efficiency, we're not measuring directly, so at best we would have to rely on models that are out there or indirect measurements. So that's not something that we have planned. Daniel.
Yeah, I was wondering, what was your sample structure like? So you had, a rye field with rye cover crop and one without. And I was wondering, how did you characterize that field? So what was your sampling structure like?
So to clarify, it wasn't that it was a field of rye cover crops and not without. Let me just go back a little bit because this might help.
Yeah.
Okay. So our study had two different fields. You see one of the fields here and then one of the fields here. Within each of those fields, there are different plots that have cover crops and don't have cover crops. So the purpose of the two fields is to make sure that each of the main crops is represented each year. Right, so you can see that we have soybeans in this field, and this field is corn. The next year, that will be opposite, and then it will go back and forth. So it's not that we have cover crops in one field and no cover crops in the other field. Within each field, we have plots where there are cover crops and then not cover crop treatments. And those are randomized.
Okay.
Does that answer your question, or?
Sure. I think we can talk about this later then.
Okay. Okay. Dr. Horton.
My question is related to Dr. Laird's question. And I'm wondering about just the way that you framed your presentation today and on looking at plant available water. Because you're really, if I understand it properly, you're focusing on taking some undisturbed soil cores, measuring water retention curves, and then defining out the plant available, the water holding capacity, the water holding capability of plant available water holding by the soil.
Yeah.
But that doesn't really address that one question that you kept hearing from the farmers. Is the cover crop stealing the water from the crop? Because, I mean, that would require a different kind of a study. That would require a water balance. When is the water available? It's not just the holding. That's just looking at a soil property, but it's not looking at the perspective of the timing. When is the water available? Is there? It has the capability, perhaps, to hold different amount of water, but it doesn't say when the water is being used, when it's actually available.
Yeah.
Are you going to do any measurements in the field of looking at soil water contents or water balance, as well as looking at the soil property, water retention?
Yeah, thank you for your question, Dr. Horton. That's actually a really important point. And starting with our next sampling year, which will be 2018, so a month from now, then I do want to put out some soil water sensors into some of these plots to get an idea of soil water content. We haven't done that thus far, but this year, 2018, we're going to put that in. Dr. Thompson.
I'm also interested in that 0 to 5 centimeter zone, which is right at the top of the soil. And during the growing season, at the beginning of the growing season, you have, I guess, rye roots are going to be in that 0 to 5. Maybe comprising significant volume of the 0 to 5. And then as those roots decompose, there will be corn roots or soybean roots that take their place. And I wonder, what is the relative water holding capacity of the roots and how that changes during the growing season? How will you collect an undisturbed sample of soil and account for the roots that can be holding the water as well as the soil pores?
Yeah, and not just the roots, but earthworm burrows. This was a headache. It really was, because sometimes I would start to take my undisturbed core and I would find there would be a big earthworm hole right in it, and I'd have to do another one. It wasn't uncommon for me to go through about 4 or 5 cores before I got one that I was satisfied didn't have too much obvious disturbance in it. So that was a major complicating factor in getting my undisturbed soil cores. As for the root growth, I did sample in the untrafficked crop interrow. So the main crop roots were not as much of a problem, but sometimes there would be some residual from the cover crop roots, and when possible I tried to take those out, but I'm sure there was still some left behind, and I need to figure out a way to account for
When you
those.
take them out, you disturb you disturb the soil, so you don't have an undisturbed
So
layer.
I should have clarified that. I attempted to take them out so I had undisturbed cores that are being used for the water retention measurements, but I also had probed soil samples that were in the proximity. So in those probed soil samples, I took those out because those are the samples that I use to process my organic carbon and particulate organic matter. So in those, the roots were taken out, but in the undisturbed, no, I didn't take out the roots because that would interfere with it.
Yeah, so my point is, you're gonna try to correlate those two measurements, and in one case, the roots will be there, in the other case, the roots will not.
Yeah.
I have one other question, and that has to do with how you will, how you'll differentiate the different types of organic matter.
Mhm.
What methods are you looking at to differentiate the slow and labile and the residual carbon?
That's a great question. There are so many different models for organic matter, so many ways to split it apart, and so many different definitions. I don't know that there's a right way to do it, but I've been basing my studies off of the century model. So I've been defining anything I define as an active soil organic matter pool is going to have a turnover between a couple of months and 2 years. Anything that's in the slow realm would be 2 to about 10 years, and anything longer than 15 or 20 years would be in my recalcitrant area. So for the slow organic matter pool, I'm using the particulate organic matter test. For the labile, then we're considering using permanganate oxidizable carbon. We're considering using that test. And for the recalcitrant, I'd like to run some humic and fulvic acid test. But there's no perfect way to do it. So many different ways. So
Yeah. I'd be glad to consult with you when we get to that.
okay, I would appreciate that. Yes.
In the beginning, you used the term study by Dr. Laird that talked about how much loss has occurred. And it was about something about 46% of the carbon.
42%, yes.
42, well, my number's wrong. Maybe the question's wrong too. But I'm concerned about how much of that 42% is subsidence. CO2, that's carbon that's in the soil that's evaporated off into the atmosphere, reducing the volume of soil that's present today versus at the beginning.
That's a great question. I have no idea.
These are
I don't know.
just side-by-side, across-the-fence row comparisons based on the top 6 inches of soil. So take it for what it's worth. To get a true assessment, you need to integrate with depth. You probably need more sites than this. But I think this gets us kind of in the ballpark of what we're talking about. But that's a good question. There's all sorts of factors that can be involved in loss of soil organic carbon.
The reason I bring that up is because I took a fence line out and you can draw in the field an aerial photo of where that fence line was because the crops are much taller in the fence line where it was as opposed to the rest of the field.
Right.
You only have to go about 3 or 4 feet away from where that fence was.
Yeah.
After how many years?
15 years.
15 years.
Yeah.
Yeah, that's a great point. Thank you. Are there any other questions?